Good afternoon. I'm Sushma Reddy from Cardiology. It's my pleasure to introduce the next session, which highlights our rising stars. These are trainees and junior faculty, and I'd say this is really one of the big highlights of this kind of symposium. These trainees and junior faculty have all received awards from MCHRI. Their work really epitomizes what is team science. So it's really not just one person, but multiple disciplines coming in together to do this work. And it also epitomizes the breadth of the work that's happening within pediatrics, all the way from machine learning to drug screening to genome editing. So I'd like to request Alan Chang, our first speaker, to come to the podium. Alan is a research scientist in the Agapur lab. Alan is a computational immunologist working with large-scale screening data sets to risk stratify neonates.
Okay, I'd like to thank the organizers for this opportunity to talk about our ongoing work, trying to define a new taxonomy for prematurity, as David mentioned earlier. And today we'll talk about how we've done so using neonatal dried blood spot metabolite profiles and deep learning. So our research program comes from a place where we're using a lot of data sources within the NICU and in the perinatal period to understand holistically risk stratification for adverse outcomes, but not just that, not just risk stratification, but increasingly how do we go beyond risk stratification to supporting clinical decision-making and interventions. So we know that historically we've categorized preterm birth into preterm, extreme preterm, and term birth in these broad gestational age-based categorizations. But even within those categories, and two individuals of very similar gestational ages can have a huge heterogeneity in outcomes, and this is shown schematically here. So what we want to do is be able to understand the risk on a more granular scale and really find data-driven ways to do that. One of the ways we've done so is by using the electronic health record. And in this published work, we've taken the various domains of events that are recorded within the electronic health record to build complex models to understand how each event affects the overall risk stratification, here shown for bronchopulmonary dysplasia. And you can see that far before we really have that outcome, we can begin to understand how the events during the peripartum period contribute to these potential risks, and ultimately, and really help our decision making with regards to that. We've also increasingly been using the notes domain here, and in this work, we've built a large language model that's trained in the neonatal domain for notes, and we fine-tuned it for downstream tasks and can do similar risk stratification. And also, importantly, this doesn't just reflect in a post hoc manner the way that the sepsis being present in the notes. We know that the model increases that probability of risk far before. And hopefully some of you got to talk to my colleague Mark about how we've also been able to approach the standardization of total parenteral nutrition through algorithmic recommendations. And that's some ongoing work that's very exciting. So today I'll talk about how we've approached this on a large population level by using a ubiquitous 55 metabolite screening panel that is performed on every newborn in the United States and in many countries worldwide. And our study question was, what other aspects of newborn risk are contained within this ubiquitous metabolite panel? Here we've used extensive efforts that predate my arrival at Stanford with the CPQCC, the California State Biobank Program, to construct two cohorts, one in California and one in Ontario, through large screening programs and the ability to link the data from administrated data sets. In univariate analyses, we found that the 55 metabolites in this panel were associated with 4 adverse outcomes of prematurity that are very grave impacts on the morbidity and mortality of premature newborns. And indeed, we built a deep learning model to be able to risk stratify these 4 adverse outcomes. And you can see in panel C that this model really captures a core set of metabolites that encompass the risk that's being expressed in this biological readout. Importantly, we performed additional subcohorting to identify those individuals for which the metabolites were more closely linked to their adverse outcomes, as seen by improved model performance. And we validated this in the Ontario cohort and also performed a similar subcohorting procedure. In panel C, you can see we've identified broadly generalizable subgroups, as seen as these individual subgroups close to this dashed line in the center. So the routinely collected metabolic screen performed in neonatal care can be leveraged for clinical insight, and multi-site collaboration is imperative not just for model validation, but in answering those new questions, while keeping in mind the site-specific requirements when we talk about broadly applying these models. The risk index offers one view into the biology of the newborn, and it may be an impactful readout for nutrition-based interventions in particular, given the link between metabolites and our TPN approach. We see the metabolic risk index as one step in this continually evolving risk snapshot in the perioperative period, drawing from data sources that are available to the clinical care team. With that, I'd like to acknowledge that many people are involved in this work and open up for brief questions.
Thank you, Alan. Very elegant work. We have time for 2 questions. I'll get started. Is it the same group of metabolites that predicted your 4 outcomes, BPD, NEC?
We think it probably is related to a general sense of metabolic maturity. I think because these 4 outcomes occur in clusters, it's pretty difficult to ascertain whether or not these core metabolites can contribute to one over another without more extensive data sets, even though our data set is very, very expansive, 13,000 extremely predetermined individuals. I think what each individual metabolite means for this requires a little bit more work, and I think is ongoing.
Okay, thank you. Since I don't see anyone else, it seems as though in many data looking at metabolites, acyl carnitine and its downstream branch chain amino acids seems to come up as being important. Was that included in your screen at all? Was that one of the 55 metabolites?
Yes, the acylcarnitines have extensive representation in this panel, and we're thinking of being able to expand what this ultimately means, because as we understand it, this metabolic panel wasn't made to capture these other adverse outcomes. They're more for inborn errors of metabolism. So one way of doing this, we think, is probably to bring in the pre-existing knowledge and broader scale metabolomics. And I know that's kind of like saying that we should just expand our view immediately. But I think we're only seeing one very small part of what that biological readout represents, and I think there's a lot more to be investigated with the biological pathways that are involved.
One last question. Oh, I was going to ask one question, but we're out of time. Thank you very much. That was well done. Our next speaker is Ying Zhu. If I could request you come up to the podium. Ying is a postdoctoral scholar in gastroenterology in the Rosen Lab. Her focus has been on chronic intestinal inflammation.
Hello, everyone. Firstly, I would like to thank MCHRI and the organizer to giving me this opportunity. And I've been learning a lot about AI this morning, and I'm excited to see how this will help our research in the future. Today, I will talk about how lipid metabolism could be a promising target in treat pediatric ulcerative colitis. Ulcerative colitis, UC, is a form of inflammatory bowel disease that causes chronic inflammation and disrupts epithelial homeostasis in the colon and rectum. It affects almost 1.25 million Americans, including 42,000 children. Unfortunately, there's no cure for UC, and the existing therapies for UC do not directly targeting epithelium. Our research focuses on understanding the diseased epithelium by studying organoids derived from colon biopsies of pediatric UC patients. Dr. Ojo from our lab did most of the work, and we are very happy for him that he's heading to an assistant professor position at the University of Alabama. His work has shown that the UC organoids exhibit impaired cell differentiation. As we can see, they have fewer buddings and differentiated cells, as well as disrupted metabolic processes, as demonstrated here by seahorse assay. So seahorse allows us to monitor metabolic activity and energy production of the cell real time by measuring their mitochondrial respiration. And on the top, here's the bioenergetic profile. We can see the UC organoids have a broad increase on oxygen consumption. And when we break it down, on the bottom, we do see a higher basal oxygen consumption rate and increased proton leak in the UC organoids. Despite this hypermetabolism, their ATP production remains at a similar level compared to the control, indicating the deranged mitochondrial metabolism and the lower energy production efficiency in the UC organoids. To better understand this, we did transcriptomics, and one of the striking findings is the upregulation of lipid metabolism pathway. And several related genes are upregulated, including fatty acid transport, apolipoproteins, sphingolipids, phospholipid biosynthesis, and lipolysis. And when we stain the neutral lipids in the organoids, we do see accumulated lipids in the UC organoids. To further investigate the altered lipids, we collaborated with Dr. Yixin Dai from CAMH across the street to perform a targeted lipidomics on a panel of more than 760 lipids. And we were able to identify 90% of the lipids from the panel in our organoids, and nearly 100 lipids are different across multiple lipid classes, and all of them were upregulated in the UC organoids. With this massive accumulation of lipids, we only see 2 free fatty acids are there, which are arachidonic acid and EPA. These 2 lipids are aligned with our transcriptomics data, as we show here. So phospholipase B1 can dissociate arachidonic acid and EPA from the membrane lipids and was upregulated. And ALOX15, which metabolizes AA and EPA to the downstream pathway, was downregulated. So these regulations may explain the accumulation of these 2 free fatty acids. 2 free fatty acid, and they might originate from membrane lipids. By studying this abnormal metabolic abnormality, we also explore the potential of correcting these disruptions by blocking fatty acid oxidation using atomoxir. As we can see with the inhibition, it normalizes the oxygen consumption in the UC organoids, and there's no difference between the UC with inhibition compared to the control group. So together with this, our study highlights the importance of targeting lipid metabolism as a novel therapeutic to treat UC. With this, I would like to all our pediatric patients, their parents and families for supporting our research, and thanks to MCHRI and for this fantastic symposium and giving us the opportunity to present. I would also like to thank our lab members and collaborators. And lastly, a little bit of advertisement. Our Center for IBD and Celiac Disease is holding our 3rd annual research symposium next Tuesday right here. We would like to invite you all come and join us. Since we have a lot of AI scientists today, we would like to seek more collaborations. And with this, thank you very much for your attention.
Thank you, Ying. Beautiful work. Could you comment on, is it mitochondrial dysfunction that's causing your lipid dysregulation, or is it the other way around?
From the data we have now, our hypothesis is the lipids are driving this mitochondrial dysfunction. Yeah, that's our hypothesis. We just got this lipidomics data pretty recently, and we're still digging into it. But the higher lipid metabolism and lipid accumulation is what we saw directly from our organoids model.
If you plan to target your lipid dysregulation as a potential therapy, do you anticipate any side effects of such a therapy?
You mean targeting the mitochondria directly?
Or if you're planning to, for example, replace the enzymes that are decreased as a result of which you think your lipids are accumulating, do you anticipate any side effects from replacing these enzymes?
Oh, thanks for asking. So all these enzymes or genes we've been identified from the transcriptomics, we didn't see any fetal enzyme that will affect most of the hub of the metabolism pathways. The data I'm showing here today, we are trying to do CRISPR knockout on some of the specific genes, and because they are not the only enzyme on that pathway, some of them are just isoforms or they have other pathways that can metabolize the molecules, so we don't anticipate any fetal or very impaired effect on those genes. We inhibit them.
I see. Well, thank you so much. I don't see any other questions, but we look forward to hearing more about the mechanisms underlying all of this lipid accumulation. Good job. Thank you. Our next speaker is Yosuke Hori, if I could request you come up. Yosuke is a clinical instructor in neurosurgery. He's trained in neurosurgical oncology and radiosurgery. His current work in the Pascal lab centers on developing an organoid model for radiation-induced brain injuries.
Okay, thank you very much for the opportunity to tell you about my work using brain organoids to identify neuroprotective components for radiation-induced brain injury. As a neurosurgeon, I care for a lot of patients with brain tumors, and then pediatric brain tumors are the most common treated tumor in the pediatric population. And then radiation therapy is one of the mainstay treatment modalities to achieve the long-term survival of the patient. But extensive radiation therapy, such as whole brain radiation therapy, is an essential part of curative treatment of the patient management. However, this extensive radiation is known to commit a significant risk to develop the long-term outcomes such as cognitive decline, memory loss, or imbalance. And the most problem is that today there is no effective treatment modality to prevent these side effects. On the scientific side, in the laboratory of Dr. Anke Pascal, I use gray matter organoids derived from the iPS cells, and then to identify novel neuroprotective components for the radiation-induced brain injury. The organoids are the small 3D groups of cells, and then that contain neuroprogenitors, neurons, and the astrocytes, which are all at risk for the injury. I used the Day-120 organoids, and then I developed an in vitro model for radiation-induced brain injury. And for this, I plated organoid in the 96-well plate, and then exposed the organoid to the various doses of the radiation between 3 and then 30 grades. And then I measured the cellular injury at multiple time points up to 48 hours after the radiation. To measure cellular injury, I use the Toxilite assay. This measures the release of adenylate kinase from the cells as a sign of membrane destruction. And using this assay, I found the dose and time-dependent injury in organoids, and then each graph represents one radiation dose. And then y-axis shows the degree of cell injury, and then x-axis shows the time point after the injury. As you can see, that there is no major cell injury between 0 to 12 hours after radiation, but there is increasing cell death at 24 and then 48 hours in all conditions. We also validated these findings using Western blot for cleaved PURP and the marker of cell death. And then given this result and the fact that 11 gray in one fraction is a similar biological effective dose of commonly used whole brain radiation therapy in the clinical setting, So we decided to use 11 gray, and then for high throughput drug screening, and then we measure toxicity at 48 hours after the radiation. For the high throughput screening, we use the Reframe library with 1,000 FDA approved compounds. So overall, I tested 440 compounds which have been shown to either anti-apoptotic, protect against DNA damage, ER stress modulators, or mitochondrial protectors. This is the summary of the screening. So we compare the extent of cell death with control samples, and then samples treated with 11 gray. And then blue line is corresponding to the control. And then this red line is from the samples with 11 gray. And then you can see, several compounds showed neuroprotective effects, and then cell death was suppressed to the control level. Next, this is a summary of positive hit compounds. So finally, we found 11 drugs that have significant cell protective effects. And then interestingly, the 2 of them are vitamin B12 derivatives. We have several other interesting compounds that are included, and we are testing too. So in this study, we established the high throughput drug screening platform for radiation-induced brain injury using human cortical brain organoid. And then a total of 11 compounds showed significant neuroprotective effects. And now we are validating this compound in vitro and planning to do in vivo validations. And thank you so much for the support from MCHRI. And then this study is supported by Clinician Educator Grant. And then thank you so much also for great support from the NeoPascal Labs.
Thank you. That was absolutely fantastic. I see you have several questions.
Great talk. The question, can you give us a hint about what the compounds were, and would you predict that are they just DNA damage repair compounds, or were they surprising to you that you found them?
I think the vitamin B12 is already reported, and then we also found several. So I'm familiar with the drugs, but it looks like interesting, and then pretty novel. So yeah, we are just validating the result.
Exciting work. One comment: there's some recent data that the neuro effects decay with time, actually, in the longer-term survival for radiation patients. There actually is partial and sometimes full recovery. Second, are your neuroprotective compounds also protective of tumor?
This is a great question. So I think some of these drugs is also protect for tumors. So I think we need to validate both in the neuron, astrocyte, and also tumor cells. And also, brain tumor has multiple types, so we need to validate with the multiple cell types or tumor types.
Thank you.
Excellent work. Thank you so much. Our next speaker is Luisa Pimentel, who is a postdoctoral scholar in medical genetics in the Gomez-Ospina lab.
Good afternoon, everyone. I'm very excited to share with you a little bit of what we have been working in Gaucher disease. So this is my work, genome editing hematopoietic stem cells as a curative approach for Gaucher disease type 1. So a little bit of background. Gaucher disease is a very complex disease with a phenotypic spectrum that goes from mild to severe based on the absence or the presence of neurological involvement. Here, I'm going to talk specifically about Gaucher type 1, which is the neurological Gaucher disease, which is one of the most common here in the Western world. Here the patients, Here the patients present enlarged spleen and liver, low blood cells counts, also present bone disease with or without pulmonary involvement, and blood malignancies. And these, And the symptoms can vary from mild to severe and can develop at any age, being childhood the most common onset. The problem here is the deficiency of glucosylceramidase, or GCAs, that leads to accumulation of its substrate, glucosylceramides and glucosylsphingosines in the lysosomes and in the tissues. This gives rise to the characteristic Gaucher cells, which are macrophages engorged with lipids that infiltrate the bone marrow and the liver and spleen, and they are considered the main factor in the disease symptoms. The standard care for this disease nowadays is include the enzyme replacement therapy and also the small molecules like substrate reduction therapy, and these must be administered every 2 weeks or every daily for the rest of the life of the patient. On the other hand, and also in the experimental side, in vivo and ex vivo gene therapy with virus or genome editing approaches has been tested through clinical trials, as well as small molecules like chaperones. An alternative, it's been known that the allogeneic hematopoietic stem cell transplantation, it's currently definitive treatment option that rapidly corrects the visceral and the bone symptoms and also can hold neurological decline in case of the other two type of this disease. But still, given its high risk of morbidity, it has been replaced mainly for the standard care. So following this idea for a one-time curative therapy for Gaucher disease, our group has proposed an approach that aims to develop an autologous product composed by the patient's CD34 or hematopoietic stem cells and progenitor cells, and modify it with CRISPR-Cas9 platforms to express the correct forms of the GBA gene, the ones that produce the GCase enzyme, in a safe CCR5 safe harbor locus. So with this approach, we were able to edit human hematopoietic stem cell progenitors and use the CD68 promoter that restrict the GBA expression only to the monocyte macrophage lineage. Here, we were able to see that this edited cell, besides expressing the GCase enzyme, we're also able to engraft in mice and differentiate into myeloid lineage in vivo. But at this point, even we were able to test all of these different behavior of the cells after editing, we weren't able to test the efficacy of this therapy in terms of correcting the disease. So for that, we decided to establish a GD1 model, mouse model, by crossing different backgrounds of mice, one with GBA with a point mutation that is very common in humans, and other background, and also with FLuxP sites, flanking a very important portion of the gene, with a MCHRI recombinase background mice. After that, we activate this CRI system, and we performing a neonatal injection of poly-IC, which acts like a mRNA, which activates our promoter, promoting the flux of these exons of the gene and creating a knockout of the GBA in all hematopoietic lineage, including the HSCs. This point mutation caused that a reduction in the GCase activity in all tissues in the mouse, so that we have a model with complete excision of the gene in the blood cells with very little, completely reduced enzyme activity in the bone marrow, and also with an early onset anemia that starts at 3 months. This model also recapitulates the visceral, resemble the visceral symptoms of the humans with spleen hepatomegalia and splenomegaly. Also accumulate macrophages and engorged macrophages in these tissues, and at 6 months of age, start to showing bone disease. So now we had a suitable and available preclinical model of GD1. So we proceed to test if our hematopoietic stem cell transplantation approach, we can halt the disease and correct the disease. So for that, we purify from these GD1 mouse, purify lineage negative cells that we expand in culture, and we edit it, this time trying to target the CD68 human promoter and the human GBA gene into the mouse ROSA 26 locus. After these, and after we confirm the target gene efficiency of this, we culture and expand the cells and transplant it in mouse, irradiate it. 4 months after the transplant of this mouse, we were able to see that the percentage of edited alleles in the bone marrow ranged from 6 to 29%. We were able to observe a normalization in the anemia in these animals. And also these transplant results in the supraphysiological GBA activity in peritoneal macrophages. Also normalizing the spleen and the liver sizes, as well as reducing drastically the Gaucher disease, the Gaucher cells infiltrated. And also we were able to detect enzyme activity in serum and in liver between 10 and 20% of the normal enzyme activity, and that was enough to normalize the lipids accumulated in the liver. And in summary, this ex vivo CRISPR-Cas9 mediated HRD can effectively restore the monocyte macrophage-specific expression of the GCase in mouse HSPCs. Our model exhibit a fast and severe GD1 phenotype. This transplantations of genome editing HSPC normalizes spleen, liver, and other hematological parameters. And finally, genome editing HSPCs can become a one-time therapy for GD1. Thank you very much. And thank you.
Thank you, Louisa. Beautiful, beautiful work, really highlighting innovation, combining so many technologies. We're out of time, so I'm going to request everyone speak with Louisa for questions that they may have. Thank you very much.
Thanks.
Could I request Zara Shokri, please come up to the podium. Zara is a postdoctoral scholar in radiology with an interest in non-invasive imaging to visualize and characterize brain tumors.
Thank you so much. Thank you, MCHRI, for giving me this opportunity for presenting my work. I'm gonna talk about my recent work about image-guided therapy of glioblastoma using theranostic nanoparticles. As you know, glioblastoma is one of the aggressive brain tumor in children and young adults, and we know there are some challenges in the treatment of glioblastoma. We focused on two major obstacles in the treatment of glioblastoma. One, glioblastoma initiating cells, which are resistant to conventional therapy, and the other is tumor blood barrier, which limits the drugs going to the tumor. For this study, we developed two theranostic nanoparticles. Theranostic nanoparticles have dual function. They combine therapeutic and diagnostic capabilities in just one platform. For this study, we developed two different theranostic nanoparticles. The first one is TMP-MMP14, which is specifically activated by MMP14, which is known to be elevated in the tumor area rather than the normal brain tissue. When they goes to the tumor and activated by MMP14, releases colchicine, which is a vascular disrupting agent, and break down the tumor blood barrier and allow the subsequent therapies go to the tumor. And the second theranostic nanoparticle is TMP-CATB, and when this TMP also specifically activated by cathepsin B, another enzyme overexpressed in the tumor, and once this TMP activated by cathepsin B releases MMAE, and MMAE is a tubulin inhibitor and effectively targeting the glioblastoma initiating cells. Both of these TMPs consist of component which is iron oxide for diagnostic purpose, and there are acts like the contrast for MRI and enable real-time monitoring of the delivery of drugs. Our purpose to show that if combination therapy of vascular disrupting theranostic nanoparticles plus tumor targeted theranostic nanoparticles are effective than just monotherapy with tumor targeted theranostic nanoparticles. For this study, we use an in vivo model by implanting GBM39 in the brain of the mice. And then we allow the tumor grow and reach the good size of the tumor to be monitored via imaging. So after that, we divided our mice to 3 groups. First group is combination therapy group. We injected TMP-MMP14, which is vascular disrupting agent, and plus TMP cathepsin B, as I mentioned, this is tumor-targeted theranostic nanoparticles. And in group 2, we just injected the tumor-targeted theranostic nanoparticles. And the last group is control group, and we just injected PBS as a placebo. As a start of the study, we did BLI and MRI as a baseline to establish the initial characteristic of the tumor. And then we start injecting our therapeutic agents. And after each injection, we did MRI, and also after 10 days for following up, we did BLI. As you see, we thoroughly characterize our drugs, and here is TMP-MMP14, and this one is TMP cathepsin B. And also we measured the size of the TMPs, and also we did some experiments for the stability of our drugs. And as you see here, over the 48 hours, in the different pH conditions, the TMPs have good stabilities. And we did MRI to show the efficacy of the theranostic nanoparticles for imaging purpose. And they were stable, and they have stable and same morphology in the TEM imaging. To ensure that theranostic the nanoparticles are getting activated in the tumor environment, we measure the MMP14 and cathepsin expression in the tumor. And as you see, the tumor's environment have higher expression of MMP14 and cathepsin B rather than the normal brain tissue. As we go to the results in the MRI, in the imaging, we have higher T2 darkening in the combination group therapy rather than monotherapy and the control group, which means we have more delivery of the therapeutic drugs into the tumor areas. And after 10 days, when we did BLI, in the combination group therapy, we have higher reduction of the signal in the BLI. But in the group 2 monotherapy group, we relatively have stable signal. And in control group, we have increase in the signal, which means we have increase in the tumor progression. We also did some histology, like Prussian blue staining, and we saw the accumulation of iron particles in the tumor, which means we have more delivery of the drugs to the tumor. And for confirming that the therapeutic drugs are more effective in the combination or monotherapy groups, we did caspase staining, and which is marker for the cell death, and we see higher expression of the caspase in the combination group therapy, which means we have enhanced cell death in the combination group therapy. And we had reduction of the fluorescence intensity in the staining of CD31, which is a marker for the endothelium cells. We saw the reduction in the combination group therapy, and also for CD133, which is a marker for the glioblastoma initiating cells. We had reduction in the fluorescence, which means that was more effective in the treatment of the combination therapy treatment. All in all, we can say the combination therapy is more effective than monotherapy in the treatment of glioblastoma. I would like to thank my PI, Dr. Dalubing, and the NIH for supporting of this study.
Thank you, Zara. That's very elegant work. As somebody who doesn't work in the brain area at all, can you comment on whether your theranostics need to be modified in any way to cross the human blood-brain barrier compared to the mouse?
Yeah, we are working in different types of theranostic nanoparticles. For example, in recent work, we are trying to attach collagenase because we have collagenase around the tumor as well, other than just MMP14 and also cathepsin B. There are some kind of enzymes around the tumor, which is making the drugs so specific for the tumors. But also we have condensed collagenase around the tumor, so we are trying to target collagenase as well. And there are different, we can modify the theranostic nanoparticles in different way, but we are now working in this kind of enzyme and also collagenase.
Right. Thank you. I think we may have a question.
Very nice work. As you know, the standard treatment of glioblastomas involves surgery, radiation, chemotherapy, and alternating current fields. Have you looked at these combinations along with your two agents?
Yeah, before we tried radiation therapy as well, before using these theranostic nanoparticles. For this study, we didn't use radiation therapy, but for the future study, we are planning to use radiation because, you know, the standard therapy is using also radiation therapy and surgery. But for surgery, the mice, we don't do surgery for the mice, but radiation therapy is one of our goals to use.
It'd be interesting to see how they all work in combination, because we really made very little progress.
Yeah.
Well, please join me in congratulating our speakers, as well as all our poster presenters. We now have a break until 2:35, so we'll see you back at 2:35.